In biotechnology and its applications, we are reading about case study number 1, which relates to BT cotton. Now, in BT cotton, what exactly happens? How do you develop a sort of a genetically modified plant? We are looking into the entire processes of what happens and how do you develop a sort of a BT cotton plant. Now, BT primarily stands for Bacillus thuringiensis. Bacillus thuringiensis is a gram-positive soil bacteria. It is a gram-positive bacteria. It is a gram-positive soil bacteria and which is commonly used by genetic engineering. It is used directly as a sort of a biopesticide. So, a microorganism which is used as a biopesticide, example is Bacillus thuringiensis. So, Bacillus thuringiensis is a soil bacteria and it is also used as a biopesticide. Now, normally the Bacillus thuringiensis occurs naturally in the gut of a caterpillar. When you take caterpillar as an insect, within the gut of the caterpillar, Bacillus thuringiensis is normally natural, it has got a natural occurrence. So, scientists normally exploit the natural situation. We exactly look in terms of what are all the characters. So, when we take Bacillus thuringiensis and when scientists studied Bacillus thuringiensis, they found it produces proteins. They found it produces proteins. These proteins are just like crystals, and these proteins or crystals are specific to certain insects only. They show high specificity. The proteins are highly specific to certain groups of insects. So, they show specificity to certain insects, not all the insects. That is why Bacillus thuringiensis has got limited applications in terms of as a sort of a biopesticide. When you take the sort of a BT, Bacillus thuringiensis, which produces the proteins which show specificity, we understand that BT is insects group specific. It can primarily cause toxicity. The BT can cause toxicity, or it can kill, it can be lethal to specific insects because it is rich in endotoxins. The BT can produce toxins or endotoxins, and these toxins or endotoxins, which are very, very specific, are applicable to 3 important insect groups. The first group are termed as lepidopterans. Now, when you take lepidopterans include the tobacco bollworm, and also the armyworm. We got 2 important examples under lepidopterans, the tobacco bollworm and also the armyworm, or it is also termed as, we have lepidopterans. Next, we have got the coleopterans. The coleopterans include the beetles, and the dipterans include the flies and the mosquitoes. So, they are very, very species specific in terms of lepidopterans or coleopterans or diptopterans. Now, when the proteins, the Bacillus thuringiensis along with its sort of proteins which are in the form of crystals, when they go and if they want to primarily kill the sort of an insect, these crystals or the proteins, they contain the sort of a toxic content. They have the toxic principle. So, the toxic principle which is present in the proteins or crystals are species specific. Now, crystals of BT toxin are produced by the bacteria. Crystals of the BT toxin are produced by the bacteria. When are they produced? They are produced primarily during sporulation. When the bacteria reproduces, when it produces the spores, this is the time when the crystals of the BT toxin are produced. And what is the other name for these sort of toxins? The toxins are termed as endotoxins. They are primarily termed as endotoxins. And what happens to the endotoxins? The endotoxins are insecticidal in nature. they are insecticidal in nature. That is, these chemicals do not kill the bacteria themselves. Many of the time you would be wondering whether when a bacteria produces toxins, why can't the bacteria itself be killed? The bacteria produces a sort of a toxins, but it does not kill the sort of a bacteria themselves, because these particular toxins are always present in an inactive state in the bacteria. The proteins are present in an inactive stage, and the toxin is inactive. And what is the toxin called as? It is termed as prototoxin. So, they are inactive and they are called as prototoxins, and thereby they do not kill the sort of bacteria itself. And these prototoxins, the Bt toxins, exist naturally in the bacteria. So, it exists naturally in the bacteria in the form of inactive prototoxins. And when insects, like when you take the leptopterans or your coleopterans or the dipterans, they ingest the sort of a toxin. That is, they ingest it in the inactive form. So, when insects taken as food, they ingest the sort of a larvae. They ingest and the larvae of the insects eat the sort of a proteins. The larvae of the insects eat the proteins, and these proteins are coded by a group of genes which are termed as cry genes, because you know the meaning of the term cry is crystal. They are coded by cry proteins. So, the larvae of the insects eat the cry proteins or the cry toxins. They are otherwise termed as cry proteins or the cry toxins. Now, when the larvae are taken in, what exactly happens? The crystals are denatured. That is, they from inactive state they go into some other state. You know about proteins, you know primary structure, secondary structure and tertiary structure. So, the toxins are solubilized in the insect gut. Their toxins are solubilized in the insect gut, and once they are in the insect gut, that is, they are present in the digestive tract. Once they are present in the digestive tract, the most important thing is they are activated under alkaline pH. They are activated due to alkaline pH. The active toxin, we have the sort of an active toxin. So, from inactive toxin to active toxin, the conversion is in the presence of an alkaline pH. It becomes an active toxin. So, the active toxin binds to the sort of a midgut. It binds to the sort of a midgut of the sort of an insect. So, once it binds to the sort of a midgut, then what actually happens? It binds to the midgut epithelial cell. The active protein binds to the midgut epithelial cells of the sort of an insect. When it binds, it creates spores. It creates spores in the epithelial cells, and it results in the swelling of the cells. It results in the swelling of the cells, and once there is swelling of the cells, there is cell lysis or cell death. There is cell lysis or cell death, and it eventually results in the death of the insect. It eventually results in the death of the insect. So, we understand if we take Bt toxin genes, the Bt toxin genes are very, very specific. They are very specific and they can be isolated from Bacillus thuringiensis, and these particular genes from Bacillus thuringiensis can also be incorporated into several crops. So, when they are incorporated into several crops, you have the development of transgenic crops. Now, the choice of the genes, the choice of whether the gene is Cry1AC or Cry2AB or Cry-AB depends upon the crop, and it also depends upon the targeted pest. The targeted pest could be leptopterans, coleopterans or dipterans. So, here we primarily have, when you take Cry1AC and Cry1AB, they are specific towards cotton bollworms. They are very, very specific for cotton bollworms, these two things, Cry1AC and Cry-AB. Whereas, if you take Cry-AB, the Cry-AB gene is specific to a sort of a corn borer. So, here we are looking in terms of what are chrygenes and how are these the genes and their sort of a specificity. They are specific to cotton bollworms as well as the cotton borer. Thereby, we have a sort of a case study of Bt cotton, wherein we are just, if you first to summarize, Bt is a very important soil bacterium. It can exist in the form of different states, and Bt primarily is it produces a toxin. The toxin is inactive. When it enters the insect gut, it becomes active under the alkaline pH and increases the toxicity. It is very, very specific to the sort of an insect, and it can kill the sort of an insect and thereby act like a sort of a biopesticide.